Hello everyone. Today I'm going to discuss about the electron and confocal microscopy, and I'm Vivek Putney. Prerequisite requirement if you want to study electron microscopy is basically you need to study first basics of microscopy. Okay, that is resolution, magnification, okay, etc. Okay, as well as the refractive index. Okay, so study those terms first and other types of microscopy first, and then go for the study of the electron microscopy. So what is electron microscopy and what is confocal microscopy? I'm just going to give you just basic introductions. Okay, this lecture is especially for biologists, okay, who want to study electron and confocal microscopy. Okay, so electron microscopy is a fantastic tool that enables the biologists to capture the images of their samples at a greater resolution than with a light microscope. Okay, and there are several types of electron microscope. Each of these can provide different information about your sample. And also the large field of electron microscope is expanding all the time, and there has been many advancements in the instrumentation of electron microscopy also. Okay, so next comes the confocal microscopy. Just a brief introduction. Confocal microscopy is basically a latest version of the microscopy which basically offers great advantages over the conventional optical microscopes. Okay, one of the main is the shallow depth of the field. Okay, elimination of out of focus glare and ability to collect serial optical sections from a thick specimens. So in the biomedical sciences, the major application of confocal microscopy basically involves either fixed or living cells and tissues that have been usually been labeled with one or more fluorescent probes. Okay, so this is the basic introduction of electron microscopy and confocal microscopy. Just a brief introduction. Okay, now moving forward with the electron microscopy. Okay, so an electron microscope, as the name itself says, the source here, instead of light, electrons will be used. The advent of electron microscope marks the significant improvement over light microscope for the study of microorganisms. The reason is very simple: better resolution and higher magnification than the light microscope. Okay, better resolution means you can clearly see the structure, and with better magnification you can see the smallest of the microbes. Okay, for example, viruses cannot be seen under the light microscopes. Okay, and the wave theory to describe the nature of electrons was given by de Broglie in the year 1924, which laid the foundation of electron optics required for the development of the electron microscopes. Okay, so in the year 1931, scientist Ernest Ruska, then 24 years old, invented the electron microscope, for which he was co-recipient of the Nobel Prize in Physics. And there are basically two types of electron microscopes. One of them is transmission electron microscope, and the second one is the scanning electron microscope. Okay, so uh, the the electron microscopes, okay, have a higher magnification power and a better resolution as compared to the other light microscopes. Okay, the using the form of electromagnetic radiation, which a smaller wavelength increases the resolution, which can be increased. Okay, whereas light microscopes can achieve maximum resolution of 200 nanometer. Okay, so this is the reason why electron microscope has a better resolution. Okay, yes, I told you electrons are used instead of light, due to which we achieve higher or a better resolution. Okay, as you know, electrons are negatively charged subatomic particles that have a wavelength, and wavelength of an electron is determined by de Broglie wavelength and is linked to the accelerating voltage used to form an electron beam. Okay, and the electron microscope provides tremendous useful magnification because of such much high resolution and obtainable with just extremely short wavelength of the electron beam used to magnify the specimen. Okay, so now let me give you something, uh, some information regarding the instrumentation of the electron microscopy. Okay, so there are several different types, as I told you. So right now I'm going to discuss two types: transmission electron microscopy and scanning electron microscopy. Okay, so you can see clear the light microscopy, TEM, transmission electron microscopy, and the scanning electron microscopy. Here you can see the differences. Now the light source here is used in the case of light microscopy, electron source in the case of electron microscopy. Next comes condenser lens. In the scanning and TM there are of special types. Okay, there are special types. Okay, tissue, or let's say specimen. Okay, specimen is placed here in the light. Same is with the condition of the transmission. Okay, but in the case of scanning electron microscopy, a scanning called a beam deflector is used, and in the case of light microscopy, the objective lens is placed here, whereas transmission is placed here, and instead of objective here the condenser lens is used in scanning electron microscopy. Okay, then comes the eyepiece lens. Okay, in light microscopy, projection lens, in transmission electron microscopy. Instead, now here comes the specimen in the case of scanning electron microscopy. Okay, now here you will be able to see the image through the eyepiece and image here through the projection lens, and the three-dimensional structure in the case of a scanning electron microscopy. But the main difference between light and electron microscopy is that light microscopy, in which air is used as a medium, but in this case vacuum, the chambers are vacuum packed. Okay, because you are going to use the electrons to check out the image, okay, of the your specimens. Okay, now let us zoom. Okay, now here is somewhat photograph of a specimen in the light microscope, transmission microscope. Okay, and this one in the scanning microscope. So what is the basic difference between the transmission electron microscopy and the scanning electron microscopy, which I'm going to discuss now? But the viewers who have viewed this video, I request you, if you want to view the complete video, please download the Science Edutech app. Okay, Science Edutech app, and purchase the course Basics of Microbiology. Okay, and the link of the Science Edutech app is given in the description. And also please subscribe the channel and share the video, as well as share the Science Edutech app with your friends and other connections. Okay,